Hello, my name is Sofía Franco. I'm in third semester of medicine in the Universidad Pontificia Bolivariana, and today I'm going to do a seminar about the article Neuroglobin alleviates the neurotoxicity of sevoflurane to fetal rats by inhibiting neuroinflammation and affecting microglial polarization. So, to begin, I want to explain what sevoflurane is. It is a halogenated inhalational anesthetic which is used to induce and maintain general anesthesia in pediatric and adult patients. Some of its effects are hypnosis, amnesia, analgesia, akinesia, and autonomic blockage. The problem of sevoflurane is that it can cause neurotoxicity. It is known that the exposure to sevoflurane during the fetal or neonatal age will later on affect the behavior, causing learning deficits and problems in the acquisition of spatial memory. Some studies have shown that sevoflurane induces neuronal death in the cortex of the hippocampus, but it can also causes fetal brain autophagy, proliferation decline, axonal injury, and neuronal apoptosis. Due to all of this, the objective of this study is to investigate whether neurotoxicity induced by sevoflurane exposure in prenatal rats correlates with neuroinflammation and microglial polarization, and whether NGB can moderate this response. NGB, or neuroglobin, is a neuroprotective protein which exhibits anti-inflammatory effects. So, methods. Pregnant rats were divided in three groups: the control, the sevoflurane, and the sevoflurane plus HGB group. And after that, they were exposed to the anesthesia. So, on the 20th day of gestation, the rats were exposed to 3.5% sevoflurane for 2 hours. And the HGB group got an intraperitoneal hemine injection 1 hour before the exposure to sevoflurane. To clarify, hemine is an inducer of the HGB. Then it was the extraction of the hippocampal and whole brain from the offspring. So, hippocampal tissue of the control and sevo group were taken on postnatal day 0, 1, and 3. And then hippocampal tissue and the whole brain were taken from the control sevo and sevo plus hemine group on postnatal day 0. Then they did a Western blot, and this test has two basic principles. The first one is the protein electrophoresis, which separates proteins on basis of shape, size, and charge, and then the indirect ELISA. On this test, specific antibodies bind to a target antigen and detects its presence and quantity. The Western blot was used in this study to detect the changes in inflammatory cytokines and microglial polarization. And then they did the immunofluorescence. This test evaluates the reaction between an antigen and an antibody, and the marker that tells if there was or not a reaction is called a fluorochrome. In this case, they used DAPY. The immunofluorescence was used in this study to detect the expression of NGB in the hippocampal tissue. Then the results. So the Western blot showed that compared to the control group, the pro-inflammatory cytokines, interleukin 6, 8, 1 beta, and TNF alpha were increased in the SIBO group. Then here we can see when they examine the arginase 1 and cluster of differentiation 163, which are phenotypic markers of M2 microglia. The response of this type of microglia mediates anti-inflammatory and can alleviate nerve injury. As we can see in these both images, the concentration of these markers increase in the sevo and hemine group. Meanwhile, the phenotypic marker of M1 microglia is the inducible nitric oxide synthase. This one is a pro-inflammatory mediator and only was raised in the SIBO group. Then we have the immunofluorescence, which shows the response of the cells to the sevoflurane. The greener it is, the more polarization by M1 microglia there is, along with neuroinflammation and pro-inflammatory markers. As we can see in this image, the more evident is the sevoflurane group. And in the sevohemine group, we can observe a marked decrease on those pro-inflammatory molecules. So, for the discussion, I'm going to compare what other authors gotten in previous investigations and what this what the results of this article were. So Shan et al. And Shan et al. Said sevoflurane exposure in prenatal rats resulted in the increased apoptosis and neurophagy in neonatal rats, leading to neuronal damage. The authors of this article agree because what was demonstrated is that the sevoflurane exposure in prenatal rats lead to a significant neuronal inflammation and cell apoptosis. Then Wynn et al. Said NGB as a neuroprotective protein responds to a sevoflurane-induced neurological damage and thus exerts a neuroprotective effect. The authors of this article agree with this statement too because the results showed that hemine increased the expression of NGB in offspring rats and that the elevated NGB inhibited the sevoflurane-induced neuroinflammation. And last, the Peng et al. And Wu et al. Said rats exposed to sevoflurane showed decreased explorative activity and increased anxiety-like behavior, as well as learning and memory impairments. The authors of this article agrees with that too, because the results showed that the exposure in prenatal rats led to a decrease in autonomic behavior, exploratory ability, and affected the long-term learning and memory. So after analyzing all of this and understanding a little bit better the uses of the molecular biology in medicine, I got two big conclusions. The first one is the molecular biology helps us to see and comprehend the effects of different medicaments and why those present, not only in the individual whom is supplied, but also in their offspring. And the second one is the different molecular biology techniques allow us to understand how different pathologies and diseases work, so we have enough elements to start an appropriate treatment. Although in this article they did not talk about a specific disease, I think this conclusion can apply to that too, because if we know what is causing the clinical manifestations of some kind of disease or problem, we can have enough elements to start an appropriate treatment.